Assalamu alaikum. My name is Samar Siddiq, roll number EBP 2001132, lab group C evening. First of all, I will discuss the classification of epithelia on the basis of number of cell layers. First of all, I will discuss a stratified epithelium. Now, what is a stratified epithelium? A stratified epithelium has multiple layers of cell. They are further classified on the basis of shape of uppermost layer. They are classified into stratified ischemic epithelium. Stratified ischemic epithelium is further classified into keratinized or non-keratinized. As the name indicates that keratinized ischemic epithelium contain keratin protein in it and they do not contain nucleus, it means that they are non-nucleated. Shape of the cell of superficial layer is flattened while the shape of basal layer is columnar. Examples are epidermis of skin, lining of parts of oral cavity. Now, what is non-keratinized ischemic epithelium? Non-keratinized ischemic epithelium do not contain keratin protein in it. They are nucleated. It means that they contain nucleus. The superficial layer is flattened while the basal layer is columnar. Examples, they lie in the oral cavity, esophagus, lower part of anal canal. Now, what is stratified cuboidal? They are round in shape. The nucleus is present in the center of the cell. Examples are, they line the duct of sweat glands. Stratified columnar, they composed of many layers of columnar cells which are elongated in shape. Examples, they line part of ductus deferens or male urethra. Transitional epithelium, it is also called urothelium. The appearance vary depending on the state of distension of organ. They may be unintended state or may be in distended state. Now, what is distended state? Distended state means that they contain or they consist of 2 to 3 layers of cell in fully distended state in which the basal cells are cuboidal whereas the superficial cells are flattened. While in undistended state contain 5 to 6 layers of cell in which the basal layers are cuboidal and can divide whereas the superficial cells are large and dome shaped. Example of distended, they lie renal pelvis, ureter, urinary gland, bladder and the part of urethra. Now I will discuss that how we make a slide of epithelial cell. In order to make a slide, first of all we take the sample and add some chemicals to it. The reason we add chemical to a slide is that we want to preserve it or to protect it from harmful bacteria or microorganism. After doing this, we will do fixation. It is a process by which the specimen tissue is prevented from being altered or damaged. The chemical we use in this process is called fixative. Fixative help in such a way that it preserves the structure of the cell by stabilizing them or by inactivating them in such a way that it unable to do autolysis. After doing fixation, we do embedding. In this method, a material is used in such a way that it surrounds or it covers the specimen. The material we mostly use is wax or paraffin. The material helps to facilitate the cutting tissue pieces into thin slices. After embedding, we do sectioning. In this process, we cut the specimen into thin slices and processed further for staining. Now, before staining, we remove paraffin by the help of xylene or benzene. Before staining, we also remove xylene or benzene by the help of alcohol and then we washed it by the help of distilled water. The process of removing the paraffin from the stain is known as deparifination. After deparifination, we do staining. In biology, the stain are usually neutral solid having acidic or basic radical and the stain is said to be acidic or basic stain on the basis of radical it has. Most common stain we use nowadays are homotoxylene or eosin. Thank you.